Hello everyone, Sanket here. Welcome back to another video and another tutorial on my channel. Today we are going to study the Ramachandran plot, and we are also going to generate this Ramachandran plot using some frequently used and widely accepted online plot servers. So basically in 1963 it was G.N. Ramchandran, C. Ramakrishnan and V. Sasisekharan who actually came up with this Ramchandran plot. Now, why did they actually think of making this thing? So what is the whole purpose of doing this plot? It was basically to help scientists and researchers in order to determine which torsion angles in proteins are permitted, and also to give a very good insight into the structure of the peptides. So that is why this concept or the funda of the Ramchandran plot came into being. Okay, so before even we go into what this plot is, we really need to understand what is a peptide bond. So each and every protein has about 20 amino acids, and these 20 amino acids are joined to each other by these peptide bonds. Now, there are two important torsion angles which we should take into consideration. You have the phi angle, which is this between C alpha and N, and then you have the psi angle between the C alpha and C. Now, both of these torsion angles or dihedral angles, what we say, they determine the geometry of the protein. Now, if these angles change, or when these angles change, they change the rotation, okay, of the bonds. So as these angles change, the rotations of the bonds will change, and if the rotation of the bonds will change, the protein will also change. That means we can say that the phi and the psi angles are responsible for the protein conformation. So each and every structure of the protein or a specific conformation of the protein is because of these phi and psi angles rotation. Now let us take into consideration, if we have one amino acid, okay, whose between C alpha, N and C, okay, the phi and the psi angles are rotated in such a way that the angle between them, so this is your phi, this is your psi, so the angle they are rotated in such a way that the angle between them is 60 degrees Celsius, which is which, let us say in this case, is normal. And there is not a lot of rotation which takes place between them, and that is why the angle it's between both of them, okay, remains at 60. Maximum it fluctuates to 62 63 degrees. Then the protein will remain to be stable. Okay, so there's not a lot of fluctuation between phi and psi angles, so that the angle between both of them, it remains to be 60 degrees, so the protein remains stable. What if there is a big high fluctuation of your phi and the psi angles? Okay, so much so that your angle, okay, it increases. And if your angle increases more than the basic normal, which is 60 in this case, okay, it's a hypothetical case of 60, then what is going to happen is your protein is going to be unstable, or in other words we can say your protein will start denaturing, because it has increased the angle between them. So the whole point of explaining this is that every uh, every rotation there is a particular limit to it. The protein does not change its rotation, does not change its the bonds do not change its rotation, the angles do not change according to their own will. There is a specific number to it, and based on that there is the rotation or the changes which take place. If there is more amount of rotation, then it leads to denaturation. So we can actually say that each and every rotation of amino acids, it helps to form a conformation of the protein. And if your conformation of the protein is absolutely correct or spot on, that means it becomes a fully three-dimensional functional protein. Okay, so if your conformation is perfect, the function of the protein will be perfect. So indirectly, your phi and psi angles, okay, they are responsible for the protein functions. We can say it in such a way. All right. Now, this is how your Ramchandran plot looks like. Okay. You must have seen this I've taken from a Google image, and you must have seen many images of this, like of this Ramchandran plot. Um, Now, this Ramchandran plot is basically nothing but like a graph which has its x-axis and the y-axis. Now, looking at this, you might get confused, like where is the x, where is the y? Okay, so let me just simplify. Okay, now this is the same plot which is there, just removed a few things. So this is your 0 to plus 180, which is plus psi, 0 to minus 180 minus psi, and this is 0 to minus 180, this is minus phi, and 0 to plus 180, which is plus phi. Now all these four things are, consider this as small graphs. So this is one graph, second graph, third graph, fourth graph. In other words, let's call them as quadrants. Okay, now I think it's much more clear. So it's 0 to 180, minus 180 to 0. Yeah, it's not like your typical graph. Okay, there's a slight thing to it, but this is your plus and this region is your minus. Your x here is your minus, your y here is your plus. So this is your first quadrant, which is your x and y. So your phi angles are negative in here, your psi angles are positive. In this case, guess this is your second quadrant. So it is 0 to plus 180, 0 to plus 180, that is psi plus phi plus. So it will be, yeah, plus plus, correct. Okay, now for this one, your third quadrant, what will it be? Okay, it's easy to guess now. All right, so your third quadrant is correct. It is minus and minus, because you have your phi as negative and your psi as negative. Okay, so these are your degree of rotations, by the way, the negative degree of rotation, positive degrees. And last is your fourth quadrant here. So the fourth quadrant will be, yes, so your x here is phi, which is positive, and y here is minus. Just seem to just take care that here this is x and y, so it is minus and plus. In this case it is plus and minus. Okay, so these are your four quadrants. Okay, and these are the various these are the ranges of your phi and psi angles. So let us say if we have a phi and the psi angle which is plus 160 and plus 160, if that is the rotation of an amino acid, of a specific amino acid, could you tell which quadrant it would lie? Yeah, it will lie in the second quadrant, because both of them are plus. So this is how you determine. So based on how the conformations are, based on how the rotations are, it lies into different quadrants. So similarly, what Ramchandran et al., so Ramchandran, Ramakrishnan and Sasisekharan, what they did was they analyzed a large number of proteins, and then they found out your first quadrant has beta sheets. The one which is negative and negative is your right-handed alpha helices, and your third this is your second quadrant. This is your third quadrant, which is right-handed alpha helices, the third one, and this is your second one, which has the left-handed alpha helices. So your beta sheets were both. I mean, this section will have both the parallel beta sheets, the anti-parallel beta sheets. So based on how the psi and the phi angles are, the secondary structures were determined. So in this way, they were very successful in order to determine what were the torsion angles, and they could also this Ramchandran plot can also give you an insight into what is the structure of how the structure of the peptides would form. So the structure of the peptides being beta sheet, right-handed or left-handed. So this is why the Ramchandran plot is created, in order to determine what the secondary structures would be. Okay, now there are two exceptions, not exceptions, but there are two cases which I would like to highlight when we are talking about Ramchandran plot. This is the cases of these two amino acids, one being proline, second being glycine. Now you can see this as your peptide chain. What you see here, okay, so each and every each and every peptide chain or an amino acid basically has an R group to it, okay. So that R group is where you add these functional groups, and based on these functional groups different amino acids are created. So adding different functional groups to the R group gives you different amino acids. So for glycine in the R, what is added, the functional side of the functional group, what is added is H. Whereas for proline, there is a much more complex structure. Now, this thing matters a lot. When we have a complex, when we have something like a proline, which is a very complex amino acid, it affects the amino acids which is there, or it affects in this case I'll put it different way, it affects the rotatable bonds, that is your phi and the psi bonds which is there beside it. Because imagine if something is very heavy, or or it is very bulky, it won't carry out rotations because it is heavy, it can't move a lot, it can't rotate itself, it can't change itself, okay, it can't move itself that much freely. So that's why we call proline as a very bulky group. So because of this, the freedom of rotation which is there between the different amino acids, so the freedom of rotation which is there between different amino acids will not take place. So we can say proline will cause something called as steric hindrance to the adjoining amino acids, okay. So two important terms here, it will not allow the freedom of rotation, and it will cause a steric hindrance to the to the adjoining amino acids. The other case, glycine, it has just one H. That means there is not a lot of burden. It leads a very simple life. Okay, unlike the proline, which leads a very complex life. Okay, but glycine, very simple life. So that means there is a free movement of rotation. So that is why your glycine is highly dynamic. The reason for mentioning proline and glycine is when we will be performing the Ramchandran plot, you will see that the glycine will go all over the quadrants. Now there's one thing which I have not mentioned here is your white section and the colored section. So the colored section is what we call as the allowed region. So almost all your amino acids which are predicted should be present in this allowed region. If it goes in the white region, it is what we call as the disallowed region. And that's why the fourth quadrant, if you see, there is nothing, because this entire thing is disallowed region. If any of your amino acids they lie in any of these white sections here, they are called as outliers. Now I told you the example of glycine, which is has a high freedom of high moment of rotation or high freedom of rotation. Glycine can lie anywhere on this just because it has a high freedom of rotation, and because it lies anywhere, generally, even though if it lies in the disallowed region, we cannot say that the structure is that the structure conformation is not stable, okay, or it is weak. We cannot say that just because looking at the glycine. All right, so as far as proline is concerned, you will see most of the prolines being in these regions. So these are the two unique cases of the amino acids of proline and glycine. When we'll be doing a tutorial, that time I think the examples, when we see the Ramchandran plot, you will be able to understand what I'm trying to say here. Now coming to what we are going to do today is we are going to look at now there are four tools in which you can carry out Ramchandran plot. You have Ramchandran server, you have Stan server, Mol, Probity, and Rampage. Today we'll be looking at only three. This is like a part of Molman 2. So we'll be looking at three different three different tools or three different online servers which can help us predict the Ramachandran plot. So, all right. So let's start with performing a tutorial for creating a Ramachandran plot.